is explainable AI. You know, up until recently, AI has been considered a black box. You know, we put numbers in, we get numbers out, we don't know what happens in between. And that's one of the problems of deep learning. We can't easily interrogate it because it's so complicated. So we can't easily see what's going on. So that creates problems. So what we need to do is think about how we can make AI explainable. So explainability is important. As I say, machine learning often criticized as a black box. I saw a paper in 1986 called Opening Up Neural Networks, Shining Light into the Black Box. So that's, you know, this is not a concept that's new. But it's still, you know, I've spent most of my career trying to make black boxes transparent. So data feeds into the algorithm, makes a prediction, but we don't know how it does it. That's a common challenge, problem. Oops. And the problem with unexplainable machine learning is we need to get an understanding of a system. Now, biologists are about understanding the mechanisms of biology, how biology works. If we've got a big complicated network which is not transparent, we don't gain any understanding. We don't gain the relationship between cause and effect. We also need to validate the findings of the ANN. So how can we validate that this has a positive influence on this if we don't know that it's a positive influence? Or if we don't know exactly what's happening, there might be other things happening in between. We also, if we are following good practice with making models, we have to understand where their strengths are, where their weaknesses are, where their limitations are, what conditions they work in, what conditions they don't work in. So we have to understand how they're working, what their strengths and weaknesses are. If they're not explainable, we don't get that. So the real rationale behind explainable AI is it builds trust. If we can see how it works, we know how it works, we can build trust. It mitigates risk. There's a big risk of overfitting with deep learning. If we can understand how much of that overfitting occurs, where it occurs, how we can limit it, that mitigates the risks. If we know about how the machine learning is working and is transparent, we can improve performance and we can improve accountability, because ultimately, if we're looking at drug discovery or diagnostic technology, we need to state to the FDA how the system works. We are accountable for understanding the biology. If we've got a black box, then that biology is not understandable. So this is kind of the current state, in my view. Most people, so the research I've done, especially in companies, 98% of companies are using large language models for their biological interrogation of data. They're finding features based upon LLMs. The whole world has jumped onto ChatGPT, but may have jumped from something much better. And if we look at the analogy of how LLMs can discover things from a corpus of literature. So a corpus of literature is, let's say, every medical journal there has been, or every biomedical journal, every plant science journal. So in this state, we have those things that are discovered and are in the literature. So all those published things, we have a body of knowledge, that's there's a slightly, it's not so clear maybe, but it's a yellow box at the top there. That's what we know. If we apply LLMs to that body of knowledge, we may be able to find new patterns, new interactions, new features, new associations that haven't previously been seen. Nobody can read every single paper and hold all of the knowledge in every single paper in their mind at one time to synthesize a result. An AI can. So taking all that knowledge into account, we have another block here on the iceberg, and this is what can be discoverable by using LLMs to interrogate the literature. Now, there is a problem. The literature, if you have a strong belief in it and it follows good scientific principles, then things are reviewed on their merit, not on their subject. The problem is things don't always get to the public, get into publication. If I write a paper that goes against, say, a particular hypothesis, if I get people who are reviewing that paper who have a contrary hypothesis, even if I present a very good argument, because it undermines their work, they're likely to reject that paper. There is bias, selection bias in the literature. The other problem in the literature is we don't present negative results. I looked at all these studies and I couldn't find any pattern in them. Nobody publishes that paper. The third problem with the literature is we haven't done that study yet. If you think about where we are, all you've seen about proteomic technologies, all you've seen about the advances in molecular profiling, all you've seen about how we can interrogate that data, well, that's only probably 0.01% of what's in the literature. Most of the literature is people doing t-tests on ELISAs. And that's a very reductionist way of looking at the problem. So all of the stuff that's there is pretty old tech. The LLMs look at that and bias what we find towards that. So it's my view, and I'm quite confident of that view, that there is a whole block under the iceberg, which are the de novo discoveries, the new discoveries we can make by doing proteomic, mass spectrometry, expression array, RNA-seq, single-cell RNA-seq work on samples and then interrogating that data using machine learning methods. That opens up a whole possibility of new things that can be discovered, and they're being discovered based upon evidence in specific data rather than non-specific evidence in the literature. So that's important to have that view of the current state of affairs. It's one thing after another this morning. Right, that's better, thank you. Just ran the risk of running out of power. Oh, okay, thanks. So let's consider the challenge as well of deep learning and explainability for omics data. So I've said omics data will struggle because the deep learning has lots and lots of things that predict. You know, I've shown that if you take 1,000 random numbers where there should be no pattern, I can predict a random number with about 99% accuracy with a deep learning model. So how generalizable is that? Well, it's random, so not. Shouldn't be able to predict, but I can. That's because of the sheer complexity of the deep learning model, fully connected model. If, however, I constrain that model, I can make the model work so it picks the best things which have the optimal performance. So we kind of have a yin and yang. We have the ability to predict, but if we have a good ability to predict, we get too many things. If we constrain our ability to predict, we get a better separation between the things that predict well and the things that don't predict well, which is what we want ultimately. So we will have problems in generality of models. We have problems with false discovery. We'll find things that are not real. We'll have problems with compute time. Deep learning models often require high performance computing, which can be a whole bank of hundreds of machines with hundreds of GPUs. There is a problem with redundancy. If I look at the activation energies in this network, I can take the activation energies across the population for each node, and I can regress them against one another. So a long time ago, we built a deep neural network, and we did that. And what we found was that around 90% of our deep neural network activation energies had a correlation coefficient of 0.95 or more. So basically, they're just repeating the same element again and again and again. And we actually did remove 95% of our hidden nodes, and we maintained a good performance. And the whole process speeded up around 1,000 times. So there's a 1,000-fold increase in speed. We weren't training redundant elements. And the last thing about deep neural networks is if we keep them simple, we can explain what's going on. Explainability means we can get a mathematical formula for our network. We can extract maths. We can publish a formula that shows how these things relate to this question. Other people can try that out. They just plug the numbers into the formula. We will get improved accuracy. So I'll just go over this again. Remember our approach. We have an adaptive system that takes an input, that predicts an output, that compares to what actually happens. That gives us an error. That error is used to update the system, and we keep cycling around and around and around until the network reaches convergence. So the network error drops like that for seen data. That's what we're doing. We're training to get to a lower error. Yep. And we use early stopping to get to that bottom point, and we want to stop because we don't want to get up here where the network's overfitted. By the way, Weka, if you use the Deep Learning 4J module, you can do early stopping in that. I should have mentioned that yesterday. So the goal is to create a model that's not overfitted, that has early stopping, that has the best possible performance for unseen data. Now, I've criticized deep learning, but how do we do it? How do we use machine learning in omics data? Well, our view is that instead of having one massive model that tries to encode, decode everything, our view is that we take lots and lots of tiny models in a swarm. So in a swarm, each model considers a gene on its own and a randomization of the data on its own. And that gives us a partial view of the problem when we solve that bit of the network. We then create another member of the swarm for a different gene with a different randomization, And by creating, in some cases, up to 10,000, 20,000 models in a swarm, we can start to interrogate those and see what's going on. Now, let's talk a little bit about that. So first of all, we can make a swarm. What that swarm does, and this is what I showed you in the latter part of Weka. We were making a model or multiple models on each gene in that list. So by doing that and interrogating the results, which we did in that spreadsheet, we were making a swarm of neural networks. So by doing that, for the whole transcriptome, we get a distribution of errors for unseen data. So this curve is showing how those errors are distributed. So they're ranked, first of all. So each point on this graph is a gene, or even a transcription factor, or a non-coding region. And what we're able to do is, let's say we ask a particular question, cancer, non-cancer. We work our way through a data set, testing every single gene with a seen data set and an unseen data set, and plot the performance for a gene on an unseen data set. If we plot that distribution, we get this sharp hook here. So what's in this hook is a subset of genes that are much better at predicting than the rest of the genes. So here, this subset of genes are the things that answer the question. And that, we do that for multiple repeats, so we end up with multiple curves like this. So we take a scan, if you like, of the gene transcriptome. We randomize, we take another scan of the transcriptome, we randomize, we take another scan of the transcriptome. And we build up this body of knowledge based upon this swarm of neural networks. So this is what that looks like. So we have a data set on tissue or blood or whatever. We randomize it. So we split it into training data, which we use to train the model. We have validation data, which we use to stop the training. And we have test data, which is used to test the model for that particular feature. We then apply that, assess every single gene, rank on performance, and then we get this enriched area here, what's in red. I showed it on the previous graph. So that has a membership of genes that are good at predicting answers to that question for that random view of the data. We then shuffle the data again. This is called Monte Carlo cross-validation because it involves an element of randomness, like shuffling cards or throwing dice. And we redo that process. So we have another enriched list with a different view of the data. We keep doing that. We usually do that up to 20 times. So we randomize 20 times, get a gene rank 20 times over 20 randomizations. And what we then look for is the commonalities in those members. So if the genes occur, what are the genes that occur in that sharp hook that crop up again and again and again? Do they keep coming into that hook? Are they consistently explaining the answer to that question? And what we end up finding is there shouldn't be anything in there, because if you're looking at the top, say 100, 200 out of 20,000 genes, now for top 200 out of 20,000, just keep the math simple, you've got a 1 in 1,000 chance of finding a gene in that. If you do 20 repeats and you find the same gene in all 20, the chance of that happening is 1 in 1,000 to the power of 20. So conditional probability is really tiny. It's not going to happen by random chance. Yet regularly we find multiple features coming up again and again and again, just breaking that random chance, which means if it's not random, there must be a pattern to it, which means that biology is important. So it eliminates false discovery, because for something to crop up, it has to crop up again and again and again, and it maybe has to crop up again and again over multiple data sets, because we can do that. We can integrate multiple data sets for the same question. So What does a swarm-based solution do? Well, it trains large numbers of smaller, less deep neural network models. Each model takes a different view of the data and addresses a different part of the problem. We can then integrate that and present all that together, and we can show overlap between the findings, cross-validate them. We use early stopping, regularization, and Monte Carlo cross-validation. So that means we're validating for generality in that whole process. And this is really important. So GPUs have effectively lots of compute units on them. So a GPU can have 1,000 compute units. That's like a little CPU. If you've got a deep model, what it tries to do is spread the whole model over all of those compute units. But the compute units have to talk to each other, which slows the whole process down. With this approach, we run one model on one compute unit. And we do that in parallel 1,000 times. So we can get a massive speed up in terms of how we assess these things by using a swarm spread over a GPU. To the point we can do 50 million molecular combinations an hour. So that's very quick, very fast, because of the optimized use. We consider all of the transcriptome from a single gene product. And then we work our way down and filter out and find the real things that answer the question. So we can assemble the model, we can reduce redundancy, we can increase speed, reduce overfitting, get a better understanding of features, and then through concordance reduce false discovery. So this approach is overcoming a lot of the problems of a deep learning network. So I'm now going to illustrate this swarm approach with a case study. And that case study is looking at common proliferation markers for breast cancer across multiple data sets. So we use 3 data sets in this. We use the Nottingham cohort, and this is quite an old study. So these are using expression arrays, microarrays, or gene microarrays as it's called. So we had a Nottingham cohort with 128 replicates on it. We had an Uppsala cohort of 249, and we had a Metabrick cohort of 1,980. We then validated this separately on a TCGA cohort. So TCGA is a massive repository of proteomic, transcriptomic, genetic cancer information. It was part of the Cancer Moonshot in the US. So this is how we did this. This is the flow diagram, if you like. So we have a data set. We looked at multiple indicators of proliferation. So that's grade, mitotic index, Ki67, survival, TOPO2, MIB, KIF2C. Each one of those we ran as a separate neural network and got a rank order across multiple repeats of each one of those features. We then did the same thing here with the next data set, with the next data set, with the next data set, and each time we looked at just the top 100 and top 200 to see which features were associated with proliferation features. So it's kind of interrogating the data. There's no one true definition of proliferation, but by using multiple definitions and then integrating the results, we kind of get closer to the ground truth. So we did this. These were some more detail about the markers and targets. I'm not going to go into those particularly today. These are the distribution of those features over the data sets. And this is where we get to the result. So across 3 data sets in the first instance, across 5 or more questions, looking at the top 100 and top 200, Looking at the top 100, we found 34 out of the top 100 genes came up again and again and again. If we looked at the top 200, 68 came up again and again and again. So these are consistent features that are there despite the different data sets, despite the different questions, despite the different randomizations. So why did we think this idea up? Because every idea has a seed or origin. And what we started to do, we were looking at models of different things, and we kept seeing the same genes coming up again and again and again. So we thought there must be something in this. Let's build this into a workflow to use that to our advantage. So let's have a look at some probabilities. These common genes in the top 100 across 5 questions across 3 data sets by random chance. Well, for a single gene, the probability of finding a single gene in the top 100 in 5 questions across 3 data sets is 10 to the minus 39. And that's calculated by 100 out of 47,000 to the power 5, because there's 5 questions. And that's the first data set. Then 100 out of 22,000 to the power 5 for the second data set, and 100 out of 48,000 to the power 5 for the third data set. So the same things were, this is the probability of finding a single gene in common. We found 34. So that value here can also be increased to the power 34. Now, I did that in Excel because even with a 16-bit machine, no, 64-bit machine, it rounded up to 0. So it's a value with around 100 zero. It's less than 10 to the minus 100. So that's more than there are stars in the visible sky, probably more than there are grains of sand on all the beaches in the world. So that's the chance of finding 1 grain of sand that's purple in every single grain of sand on the whole planet, or 1 grain of sand that is of particular characteristic. There probably are purple sands, but you see what the chance of it being a false discovery are so low. So that we can use to our advantage. Where these 30 or 68 genes, what did they come up in? How did they compare with what other people found? Well, there were 54 genes out of the 68 that are known to be associated with mitotic cell cycle. That's a Go process. 25 with chromosomal segregation, 33 with cell cycle, 27 with microtubule processes, 25 against protein assembly complexes. So those things are coming up. Molecular functions, tubule binding, tubule activity, adenonucleotide binding, and motor activity. So these are all about the mechanics of how a cell divides, how the cells are pulled apart. Obviously, when a cell is dividing quickly, those mechanisms are changed. And proliferation is how quickly cells are dividing. And when cells are dividing too quickly, it's more likely that you get partial separation of the nuclear material, which means that you don't get true copies of the cells, which means you get various genes mutating, and you get what's called chromosomal instability. So a lot of what we find is associated with chromosomal instability. So there are 100 genes in the SYN attractor. We found out of our 68, 53. Genomic grade, SYN 70, PAM. So we get a lot of overlap with what other people have found, but we've also got unique features. And that's important, because if we just found everything as unique in the whole list, then there's no validation there. But if we're finding things that are associated with the question as have been found by other people, that validates the new things we've found as well. So this is that same network which I showed you earlier. This is we then went on to validate this with immunohistochemistry. So these are cytoplasmic stainings with strong and moderate. This is really interesting. Oops. So this is looking at cytoplasmic expression and associated with anthracycline response. So anthracycline is a chemotherapy for breast cancer which attacks highly proliferating cells. So if we know and have an index for predicting how proliferating a set of cells are, we can use that to predict how somebody is going to respond to that chemotherapy. A personalized medicine, if you like. So what we did is we looked at outcome. And this outcome data is retrospective, but we have outcomes for 10 years in this cohort. So we looked at SPAG5 nuclear expression. And what we see in nuclear expression is low expressors tend to do better than high expressors when they're treated with anthracycline. If we look at cytoplasmic expression, then it flips. Low expressors of cytoplasmic expression do much worse than high expressors. So if SPAG5 is basically, if SPAG5 is in the cytoplasm, not the nucleus, which is where it should be, people are more likely to respond to anthracycline. So we can use that, and it's a way of understanding the disease more. And sorry, yeah.
This prediction has this been tested clinically as well, like in ER-negative breast cancer patients, if the cytoplasmic expression was high and anthracycline was more, so has it been tested clinically as well? Yes.
So this is a clinical cohort. And then we went on, you'll see in the paper in a second, we went on to validate this in 15,000 cases in a Lancet paper. So, good question there. So, yeah, we ended up validating this. We went to everybody else with their survival data, with their anthracycline treated cases, sent them a monoclonal antibody so they could stain their tissue microarrays, got the data back, and these were cases across Northern Europe, Canada, USA, New Zealand, Australia. I think there were Southeast Asia, South Asia cohorts in there as well. And then brought that all together, and we could see that actually this predicted, as we say, anthracycline chemotherapy sensitivity. And we went on to also validate it by copy number variation and protein expression as well. So with machine learning, we'd got to this new target of proliferation. In fact, based upon this paper, the Lancet said this is probably a better predictor of proliferation than the current gold standard, which is Ki67. So another way which we can use swarm-based learning is for diagnostics as well. So we can iteratively add to our network. So we pick the best single gene using our methods, and then we add everything else to it as a single gene to find the best pair, and then the best triplet, quadruplet, quintuplet. So we add and build up a panel of markers in an additive fashion. So we can say, what is the best combination of genes that predicts this question? So we pick the best single feature and then sequentially add to it. And again, we have these multiple layers of cross-validation, and we can build this into a piece of code. So what we've done here, this is a graph. We've got the sensitivity. We start off with a single gene. The sensitivity is about just over 50%, which is pretty bad at predicting on its own. And then sequentially adding to it, we can take that predictive ability up to, if we take 25, up to around 85%. So we find the best combination by sequentially assessing every pair, every triplet, quadruplet, quintuplet, sextuplet, and so on across the whole data set, across the whole transcriptome. And in this way we can start to build panels of diagnostic markers and build decision support tools. So one of the things we've done here, for example, is to look at chronic obstructive pulmonary disorder, look at a panel of markers and predict whether somebody is heading towards an exacerbation. Are they likely to have an episode of COPD? How long is that going to be before that happens? And that's just a piece of work that's just about to come out in the press, in publication. Another place where we've used this, we call it precise, is in identifying diagnostic biomarkers of latent TB. So by taking whole blood from patients, we were able to do transcriptomic profiling. We had 3 states. We had active TB, latent TB, who were symptom negative, and healthy. So we took the data, and with 4 markers, we had an AUC of 0.95. So we had 98% sensitivity, 88% specificity. So we were able to, again, optimize a diagnostic panel. Discriminating control versus latent, we could also discriminate control versus active and active versus latent. And then we also modeled, using the network inference, the transition between latent TB and active TB. And we could start to see switches that were coming on when people were moving towards active disease. We then worked and developed this and validated it in a small Chinese cohort, 50 cases. This was with a hospital which people hadn't heard of until recently. It was in Wuhan, in China, Wuhan Pulmonary Hospital. We started our trial in 2019. Halfway through our trial, we had COVID hitting. It was a pulmonary hospital, so needless to say, those people were impacted, and sadly, some of those people in the clinical team died from COVID during the course of that study. So that's quite a sad state from this trial. Nevertheless, the result worked, and hopefully we can bring a diagnostic that helps people in TB. So this is patented, and we're now talking to a large diagnostics company about taking this panel and the algorithm forward as a way of diagnosing large-scale populations for TB. Another area we've applied this is Alzheimer's disease. So in Alzheimer's you have healthy, you have post-mortem confirmed Alzheimer's disease, and you have mild Alzheimer's, which is called mild cognitive impairment. So the healthy really is a misnomer, because this is brain material from dead patients' post-mortem samples. So I don't know, we shouldn't really use the word healthy because they have died. They're not healthy. But they are controls. They were confirmed not to have AD when they died. So what can we do here? The idea is to understand the mechanisms of disease, to build diagnostic methods that help us ultimately go towards blood, where we can build a diagnostic test for blood. But the data was out there. We used it using 5 markers. We ended up with a training performance, so it predicted 94.85% accurately. So it was tissue material. It took us 4 weeks to discover it, and we found a 4-gene panel. Actually, it was a 5-gene panel. And we had sensitivity of 96%, specificity of 94%. What's interesting here is we plot the probability distribution of what is the probability of an individual having Alzheimer's? So we get this big block of individuals up here, which are clear, high probability Alzheimer's cases. And then we get a few individuals just starting to look more like healthy controls. Likewise, all this block here are controls. There's a few controls which are false positives. So they don't have Alzheimer's, or they were not diagnosed with Alzheimer's, but they might be heading towards Alzheimer's. And then we applied the blind mild cognitive impairment cases into the mix. And most of those, with the exception of one here, most of those fell between disease and healthy. So they were more like Alzheimer's, but they weren't as probable as having Alzheimer's. Excuse me. So that's ending. Next lecture. Any questions? So this is how we can use machine learning, omics data, proteomics, transcriptomics, blood, tissue, how we can use this data for diagnostic purposes and build diagnostic decision support tools. So any questions? Yeah.
So you told us about regarding this way for the reduction of risk of false discovery. So how much it is effective in reducing the false discovery?
So could you speak up a bit? I can't hear you. I've got a fan just here. Do you want some microphone?
You told us about the genomic distiller for the reduction of the risk of false discovery.
Yes.
So how much it is effective in reducing the false discovery, and with respect to the DIA and DDA, how would it be differentiated?
Right, so yeah, it's changing the probability of false discovery by giving us effectively a calculated value. So what is the calculated false discovery rate if we look at a deep learning model? Well, we don't really estimate that, but what we would have to do is compare what we found to a distribution. Or what we found 200 genes of import out of the whole transcriptome. That would give us a probability. We then compare that to the probability we get with this new method, and we see a big decrease in the probability. So it's changing the probability of false discovery by quantifying it and allowing us to be very selective. We don't get that quantification in the deep learning model. In terms of DIA and DDA, that's a different false discovery. Has the gene been identified correctly by the mass spectrometry instrument? What we're saying here is the gene or protein, so it's protein, gene for DIA, DDA, is this protein or gene, is it really associated with that question? So it's two different terms, same term, but different contexts. So for proteomics, is it a real protein that we've found? Does it exist as a real protein? Here does it exist as something that really predicts an answer to the question? So that's where the difference lies. Okay. Any other questions?
Sir, in your swarm models, if suppose we have different biological study entities, like for location or something like different. So that different models will be specific to that entities, or it will incorporate all entities and randomly selecting them?
If we've got data for the same individuals, like proteomic, transcriptomic, genetic, well, then we can mine all of those together. So we can allow the genes, the proteins, the transcripts to compete with one another to find their best combination that predicts an answer. So as long as it's in the same data set, it doesn't matter. We can also, if we've got spatial data, we can tag location as well as MZ value, or location as well as gene identity, together, and then mine that as a spatial question. So we collapse the whole spatial data into a single 1D list, but by tagging the names of the location onto the names of the genes.
Suppose like it's in single RNA data we are using it.
Yeah.
So like other meta information, we'll just tag it along.
Yeah, we can tag that on, so it might be an important feature. What I would suggest is probably tagging it on and then not tagging it on and see how the results vary. Because if you've got whole transcriptomic data, then in theory that all together should explain the answer to the question. If, however, you've got other variables that are explaining the answer to the question.
Yeah, suppose like other RNAs, microRNAs.
Yeah, we can include, well, we do, when we do a deeper analysis, we'll include all the non-coding, all the microRNAs, all the transcription factors anyway.
And what is the probability if we get a small set of data as compared to the general AI size data? Like in small cohorts or something like that?
Yeah, so small cohorts, because we have an independent framework for assessing the probability of false discovery, what we tend to see is if we've got lower numbers, then it becomes a bit more variable. Now, the pool of things that are in that stable region drops because we're not actually, we don't have a full or better, such good representation of the system. Whereas if we've got around a couple of hundred individuals, then we get really good stability results. Out of the top 500, the best I've seen is 460 features out of the top 500, consistently predicting the answer to the question across 20 different runs of the model. So that's pretty stable. Sometimes we might not find stable things, and we've got lower numbers. It might be as low as 20 or 30, so we have to bear that in mind. But we can, with this, compare the quality of results from multiple data sets as well. So say we get 3 data sets. The first data set we get 250 features, second data set we get 180, third data set we get 300. By looking at each